The following video will cover subtopic 1.4, membrane transport. The following are the understandings, application, and skills this video will cover. So in the previous video, we covered that the phospholipids within a cell membrane will form a bilayer. The function of that phospholipid bilayer is that it will form a barrier. But it turns out that that barrier is going to be selectively permeable. So what does this mean? So what selectively permeable means is that some molecules will pass through easily in this process of diffusion, which we will cover, or they can pass through the membrane using a tunnel, referred to as facilitated diffusion. Some molecules need energy to get them through the cell in a process referred to as active transport. And then some molecules are too big to get through a tunnel or to go through by simple diffusion. So the cells will have to use their own membrane to get them through in a process referred to as endocytosis or exocytosis. So starting with these ways in which cells get things in and out, we'll start with the process of endocytosis and exocytosis. So in endocytosis, you can think of this as a substance entering a cell. So in that process of endocytosis, a substance is going to be too big to actually pass through the membrane. So the cell will now reshape its membrane to bring that substance into the cell itself. So to do that, the cell will form a vesicle on the inside of the plasma membrane, and this will allow materials to be taken into the cell. So sometimes these vesicles will contain water and solutes from the outside of the cell, but oftentimes they will contain larger molecules needed by the cell that cannot pass through the plasma membrane. So the process in which this happens is that a small region of the membrane is pulled from the rest of the membrane and is then going to be pinched off. So this is illustrated using the following diagram. So if this is our substance that is going to enter the cell, and it's far too large to pass through the plasma membrane, as the substance approaches the cell membrane will pull back and will basically kind of fold on itself now to allow the substance kind of closer, start drawing that substance into the cell. As the substance is being drawn into the cell membrane will now move upwards and start to fold over onto that substance. Eventually, those two parts of the cell membrane can join together now, as you can see here, forming a vesicle around that substance. So this substance is now inside the cell. And this will form a vesicle which is known as an endosome, but for us we'll just refer to it as a vesicle. And this vesicle at this point can now go off to be combined with a lysosome to try and break down those large molecules into smaller molecules within it. In the process of exocytosis, it's basically going to be the opposite of endocytosis. So we can think of this process now as substances exiting the cell. So these are substances that are going to be too large to just travel through the plasma membrane, or not efficiently allow them, wouldn't be efficiently moved through a plasma membrane. So in this case, with exocytosis, you're going to have a vesicle that fuses with the plasma membrane and then will release its contents outside of the cell. So in the diagram, it demonstrates this process of exocytosis. So if we have a secretory vesicle that is, has contents to be released to the outside of the cell. So If we consider this the inside of the cell, and this the outside of the cell, that vesicle is going to move towards the plasma membrane, and the membrane of the vesicle will start to fuse or merge with the phospholipids of the plasma membrane. Remember, those phospholipids are fluid, they can move about. So you can have the two membranes essentially join together and allow those phospholipids now to interact with each other. As the two membranes fuse, this will now create an opening that will allow the contents of that vesicle to be released to the outside of the cell. So these vesicles are very useful for releasing these materials from the cell. After proteins are made in the rough endoplasmic reticulum and processed in the Golgi, they can then be released. So where exocytosis is also useful is that it can help expel waste products or unwanted material from various cellular metabolism reactions. So there are other ways that the cell will get materials in and out or move them across that cell membrane. These include the process of simple diffusion, facilitated diffusion, osmosis, and active transport. In the first process, which is simple diffusion, and understanding simple diffusion will set up our understanding for the other processes of getting stuff in and out of the cell. So with simple diffusion, so if we just talk about diffusion, diffusion itself is the passive net movement of particles from an area of high concentration to an area of low concentration. So what that means is if you have substances crowded together, this would be an area of high concentration. Over time and naturally, without any energy input, they will eventually move out and find areas of low concentration. So you can think of there are plenty of examples of this all around us. So for example, when somebody sprays Axe at the back of the classroom, initially you have an area of high concentration of those Axe particles, and then they will naturally spread about to areas of low concentration, eventually moving from the back of the room to the front of the room, and eventually everybody in the room can smell that Axe scent. And so when substances move from an area of high concentration to low concentration, we consider this substance to be moving down its concentration gradient. Now, oftentimes when we deal with diffusion, we are dealing with a semi-permeable membrane, so we get diffusion through that semi-permeable membrane. And this is a process that does not require energy in order to occur. So this is an energetically favorable process. So with diffusion, as the diagram shows, is that if you have a high concentration on one side of the membrane and a lower concentration on another, the overall movement of these particles is going to be from an area of high concentration to an area of low concentration. And if you have a higher concentration of substances on one side of the membrane than on the other, you are going to get very fast diffusion across that membrane. Whereas if you have slightly higher concentration on one side of the membrane as opposed to the other, so in this diagram we have 1, 2, 3, 4 red circles. We have 1, 2, 3 red circles. So on the left side of the membrane, we have slightly more red circles than on the left. And so, you will get a net movement of particles now going to the left, but it will be at a much slower rate because there is not such a steep difference in concentrations on either side of the membrane. So, using this idea of diffusion now, this can be applied to facilitated diffusion. So, when you have ions and other particles that cannot move between the phospholipids, or can pass in and out, they can pass in and out of cells if there is a channel for them. And this is the idea of facilitated diffusion. So these channels are going to be proteins, and they're going to be the integral proteins that were discussed in the previous video. And those integral proteins are going to have a channel in them. Basically, you're going to have a narrow hole down the middle. And so the diameter and the chemical properties ensure only one type of particle can pass through. So for example, you can have a sodium channel or a potassium ion channel, but not both in facilitated diffusion. So the size of those ions would preclude one from using, would preclude both of them from using the same channel. So the following diagram goes through the difference between diffusion and facilitated diffusion. So in simple diffusion, you simply just have particles or substances traveling through the membrane and going from an area of high concentration to an area of low concentration. In facilitated diffusion, these are substances now that are going to be too large to directly pass through the membrane. So you will have an integral protein, which acts as a protein channel that will allow these substances to pass through the membrane, and they will be specific to that one molecule. So these protein channels now will allow these molecules to pass through the membrane from an area of high concentration to an area of low concentration. And they can have some shapes to them as well, so they can be, they can have a hole directly through the middle of them, through which substances can travel, or they can change shape, that allowing substances to pass from the outside of the cell to the inside of the cell, or vice versa. There is a special type of diffusion referred to as osmosis. Osmosis refers directly to water movement. So water is able to move in and out of most cells freely, so it can travel across that semi-permeable membrane relatively easily. And there's channels that will allow that to happen. So sometimes the number of water molecules moving in and out of the cell is the same, and there is going to be no net movement of water. So you have equal concentrations of water inside the cell and outside of the cell. Other times there are more molecules moving in one direction than the other, so you get more molecules moving out than in, and this is referred to as osmosis. So in osmosis, due to the differences in concentration of substances that are dissolved in the water, which we refer to as solutes, this will cause that movement of water from one side of the membrane to the other. So substances dissolved in water form these intermolecular bonds, meaning they just interact with water, or they interact with these water molecules. And this will essentially restrict the movement of water. So the formation of these types of bonds can prevent water from moving from one side to the other. So regions with higher solute concentrations therefore have a lower concentration of water molecules free to move. And therefore, water can move from regions with low solute concentrations to high solute concentrations. And this is a process in which no energy is needed for this to occur. So this process of osmosis is demonstrated in the following diagram. So if we start with this semi-permeable membrane where we have equal volume of water, on one side of the membrane we have a high concentration of solutes, or just substances dissolved in water. On the other side, we have a low concentration of substances dissolved in the water. And over time, what will happen is because there is more free water molecules on the left-hand side, and there are a lower number of free water molecules on the right-hand side, more of those free water molecules are able to travel from one side to the other. And you'll get more water molecules traveling from one side than the other. And you'll eventually reach a point where the concentrations of those two solutes will be equal or equivalent on the two sides of that membrane. Osmosis is very important for a cell in terms of water moving in and out of it. So there's a lot of importance for osmotic control. So you can have several conditions in which water movement in a cell would be affected. So there are different types of solutions based on its solute concentration. There is what's referred to as isotonic solutions. These are solutions that have the same solute concentration as that of a cell. And so with an isotonic solution, you have an equivalent amount of water moving into the cell as you do moving out of the cell. So in isotonic solutions, there is no net movement of water in or out of the cell. So the cell is, able to maintain its shape and able to function normally. In solutions, in solutions now that are considered hypertonic, and this is where you have net movement of water out of a cell. This can cause the cytoplasm to shrink, and you can get cells now that are shriveled and won't function properly. You can also have hypotonic solutions. This is where you have a net movement of water inside the cell, and that net movement of water inside the cell will cause the cytoplasm to expand, and it could cause the cell to rupture or burst. And so ultimately, why do we care that it's isotonic and hypertonic and hypotonic? Basically, the salt concentration of that solution could have an effect on the health and the health and well-being of those cells. So oftentimes, saline is going to be used in IVs, and saline is considered an isotonic solution, as it is a 0.9% solution of sodium chloride that is going to be used in IVs. And it's used to rinse wounds and abrasions, keep damaged skin moistened, to use in eye drops, and keep organs fresh for transplant. So this isotonic solution will maintain the health of those cells. In the last form of cell transport, there is the process of active transport. So active transport will allow cells to take in substances even though there is a higher concentration inside than outside the cell. So active transport is a process in which substances move with their concentration gradients. So the molecular ion enters the protein pump and can basically reach as far as its central chamber. A conformational change in the protein pump, and conformational change all we mean is literally the protein will change shape. So it'll like literally change how it looks. And so that conformational change will take place using ATP, and the ion or that molecule can then pass to the opposite side of the membrane. So what active transport is going to do is it will take a molecule, for example, that is at a low concentration on one side of the cell, and move it to where it is at a high concentration on the other side of that cell. So as that molecule now enters into that protein pump, the protein has one particular shape to it, and once that ion enters, ATP is then used, and that protein pump will now change shape. And this will allow the ion to move out of the cell, for example. So there are some specific examples that Ivy would like you to know about active transport and facilitated fusion. And these both have to do with these pumps and channels found in the axons of neurons. So just as a little bit of background before we get into talking about these specific examples about neurons, is that axons are a part of a neuron. So a neuron makes up those cells as part of our nervous system. They send information in the form of electrical signals. The structure of a neuron is shown in the diagram. It's the blue arrow. And the neuron itself has a number of features to it. So there are these branch-like structures coming off of the main cell body. These branch-like structures are referred to as dendrites. There is one long extension coming off of the cell body that is referred to as the axon. And it's on the axon that we will find these channels and pumps that Ivy wants you to know. So also what neurons do is that they will function to convey messages rapidly in an electrical form, referred to as a nerve impulse. So a neuron will receive some form of stimulus that will create this electrical signal that will travel down the axon, and then it will pass the message on to an adjacent neuron through the release of neurotransmitters. So to pass on that electrical signal, the neuron will use a sodium-potassium ion pump that will follow a repeating cycle of steps that will ultimately result in 3 sodium ions being pumped out of the axon and 2 potassium ions being pumped into the axon. So 3 sodium ions being pumped out of the cell, 2 potassium ions being pumped in to the cell. And each time the pump goes around, it will use 1 ATP. So to get the 3 sodiums out and the 2 potassiums in, 1 ATP is going to be used. So what that looks like in terms of those series of steps is the following. So essentially you have the extra, so we'll set up our diagram. So we have the extracellular fluid, which we can think of as outside of the cell. We have the cytoplasm, which is inside the cell. We have our cell membrane, embedded in that cell membrane of the axon is the sodium, sodium ion, potassium ion pump. And notice that we use the word pump, which means that this is involved in active transport and uses ATP to transport substances. So in the first part of this, you'll have 3 sodium ions that will bind to the pump. And then you'll have ATP, which will be used to transport or release those 3 sodium ions to the outside of the cell. On the outside of the cell, there is going to be now, or it's going to start building up a high concentration of sodium ions on the outside. So on the inside, there will be a low concentration of sodium ions. So to get those sodium ions out, ATP is used to do that. In the next step of this process, you have these potassium ions that will bind to the pump. The pump will then change shape, releasing those potassium ions into the cytoplasm of the cell. And what you end up with on the inside of the cell is a high concentration of potassium ions and a low concentration of potassium ions on the outside. And this process will continue to repeat itself, building up this high concentration of sodium ions on the outside and high concentration of potassium ions on the inside. The example of facilitated diffusion that IB would like you to know is that of the potassium ion channel. So on the cell membrane of the neuron, that cell membrane will have a charge associated with it. So on the outside of the cell, that membrane will have a positive charge associated with it. On the inside of the cell, that side of the membrane will have a negative charge associated with it. This difference in charge is specific to neurons and to the axon in particular. And this difference in charge is referred to as a polarized membrane. Normally, membranes will have an equal charge inside and outside of the cell. But the axon will create this difference in charge through the use of that sodium and potassium ion pump. Because you have more positives on the outside than on the inside of the cell, this creates an overall positive charge on the outside of the cell, and as a result makes the inside of the cell negatively charged. So the way the potassium ion channel is going to work is that it's what's referred to as a voltage-gated channel, meaning that it's going to have a certain shape depending on the charge of the membrane. So in the potassium ion channel, and remember that we have a large concentration of potassium ions on the inside of the cell. In this configuration, in this diagram, right now this channel is considered closed, and potassium ions cannot pass through this channel. In this conformation here, there are a lot of positive charges, or a large amount of positive charges in that protein that will favorably interact with those negative charges. And this is what allows the membrane, or allows the channel to be closed. During a NERD impulse, the membrane, the charge on the membrane will now become flipped. So now the outside of the cell will now gain a negative charge, and the inside of the cell will now gain a positive charge. Those positive charges inside the potassium ion channel will now be repelled by these positive charges on the inside of the membrane and be attracted to those negative charges that are now on the outside. So this will cause a change in shape of this channel and will now allow these high concentration of potassium ions to start flowing outside of the cell. And this process happens very quickly. You get a rapid release of potassium ions out of the cell, and the cell will look to close off that process or shut that channel very quickly. So there is this additional structure to the protein referred to as the ball and chain. And in the open conformation, the ball and chain is going to be attracted to those portions of the protein channel and will move up into that channel and will close it off. And eventually the cell membrane will return to a state where it is positively charged on the outside and negatively charged on the inside. And the potassium ion channel will return to its original conformation. And this whole process will repeat itself again with the next nerve impulse that passes through that neuron. So that is it for this video for 1.4.